Good morning children, I am Sardiasis. Now I am going to teach you transcription in prokaryotes. As I told you, in prokaryotes they are polycistronic. That means in the structural gene several cistrons are there. Here the three types of RNA are formed by the enzyme DNA dependent RNA polymerase. So the enzyme used for the transcription of three types of RNA is DNA dependent RNA polymerase. And in prokaryotes we can see the transcription as three steps. The three steps are there in the transcription process. The first step is initiation. And the second step, elongation. And the third step, termination. So these are the three steps in the transcription process in prokaryotes. And now we can see one by one in detail. First step, initiation. So in initiation, the promoter gene, as I told you, the three units of transcription are promoter gene, structural gene and termination gene or terminator gene. The promoter gene can be recognized by the sigma factor of the RNA polymerase. So the sigma factor of the enzyme, which enzyme? RNA polymerase enzyme, recognize the promoter gene. Then it binds with the promoter gene. The RNA polymerase enzyme binds with the promoter gene. And that is the initiation, and that causes the unwinding of the double-stranded DNA. And one of the strands act as the template strand for transcription. So that's all about the initiation process. So what are the points in initiation? The first step is the recognition of the promoter gene by the sigma factor of the RNA polymerase enzyme. And the enzyme name is DNA dependent RNA polymerase enzyme. Okay, so after initiation, there is an unwinding of double-stranded DNA and one of the DNA strands act as the template strand. And next we can see the second step, elongation. See, after initiation, the RNA polymerase moves towards 5' 3' direction. As it moves along, new nucleotides will be added for the formation of mRNA, and that is the elongation. So before elongation, the sigma factor leave from the promoter gene or from the structural gene. Okay, so polymerization occur. Polymerization means extension or adding of nucleotides on the template strand of the DNA. Next step is, or next process is termination. See, termination, there is a terminator gene in the segment of DNA. When the RNA polymerase enzyme reaches this terminator gene, that defines the end of the transcription. And it is facilitated by a factor which is called a Rho factor. So, a Rho factor that form a hairpin loop to stop the transcription process. So, that's all about the steps involved in the process of transcription. And in prokaryotes, I told you they are polycistronic. So, no exons and introns are there. So, no need of any post-transcriptional modification. They can directly enter into the translation process immediately after the transcription. No need of any post-transcriptional modifications. Hope you understood everything about the transcription in prokaryotes. That's all for today.